Hello student, this is the chapter Cell, the Unit of Life, and this is video number 4 in continuation to video number 3. If you all have not seen the video number 1, 2, and 3, I would tell you to see that. And in this video, we'll be talking about ribosomes and nucleus. So what is a ribosome? Ribosomes are present both in prokaryotes as well as in eukaryotes. These are non-membranous granular structure composed of ribonucleic acid, that is RNA, and proteins. George Pallet first observed ribosome in 1953. In prokaryotes, you had studied that the ribosomes were of 70 S, but in eukaryotes, the ribosome is of 80 S. The two subunits of these ribosomes are 60 S and 40 S. Together, it forms 80 S. Now, what is cytoskeleton? It is a network of filamentous proteinaceous structure present in the cytoplasm. It provides mechanical support, motility, and it also maintains the shape of the cell. The next topic, cilia and flagella. We all know that cilia and flagella helps in movement. They are like structures. Cilia are small structures which works like a oar. It causes the movement of the cell or the surrounding fluid. Flagella are lower and they are responsible for cellular movement. Flagella in prokaryotes and eukaryotes are slightly different, structurally different. But if we see to the structure of the cilia of flagella, they both are covered by plasma membrane. Their core, that is the exome, has many microtubules running parallel to the long axis. Exome has 9 pairs of doublet of radially arranged peripheral microtubules and a pair of central microtubules. Thus, it is known as 9+2 arrangement. The central tubules are connected by a bridge and are enclosed by central sheath. It is connected to one of the tubules of each peripheral doublet by radial spoke because there are 9 radial spokes. The peripheral doublets are also interconnected by linkers. Now let's talk about centrosome. Centrosome also has microtubules, but it is a 9+0 arrangement. They are surrounded by pericentral material. The centrioles lie perpendicular to each other. Each centrosome is having 2 centrioles, and the centrioles lie perpendicular to each other. Like just now we saw in cilia, there is tubules in the center. It was a 9+2 arrangement. This is 9+0 arrangement. And the second difference is that the microtubules had the doublet, but here it is triplet. The central part of the centriole, it does not have a microtubule, but it has a proteinaceous structure, and it's called the hub, which is connected with the tubule of the peripheral tubule by radial spokes. That's how we develop. The centrioles form the basal body of the cilia or flagella and spindle fibers. Spindle fibers, that is the formation and a very important part of cell division in animal cell as well as in plant cell. Nucleus in the interface of the nucleus, there is a nucleomembrane, nuclear matrix, a nucleoplasm, and a nucleus. Nuclear membrane or nuclear envelope, it is a double-layered membrane with a space between 1 to 10 nanometer called the perinuclear space. It is a barrier between the materials inside the nucleus and the cytoplasm. Outer membrane usually it is continuous with the endoplasmic reticulum and bears ribosome. Nuclear envelope has minute pores, which is known as nuclear core, and that is formed by the fusion of the two membranes. These are the passage for the movement of RNA and protein between the nucleus and the cytoplasm. Inside the nucleus, there is chromatin material. These are nucleoprotein fibers. They are DNA or some basic proteins known as histone proteins, non-histone proteins, and RNA. During cell division, chromatin condenses to form chromosome. Nucleolus, it's one or more non-membranous spherical body. It's continuous with the nucleoplasm and is the site of ribosomal RNA synthesis. The human cell has about 2 meter long DNA distributed among its 46 or 23 pairs of chromosomes. Every chromosome has a primary constriction known as centromere, and on the side of which disc-shaped structure that is the kinetochore. Based on the centromere, position of the centromere, the chromosomes can be divided into metacentric, submetacentric, centric, and telocentric. Let's start first with metacentric. Metacentric chromosomes are the chromosomes where the centromere is in the middle, so that both the arms are equal in size. Submetacentric centromere is near to one end, formed such that one arm is slightly shorter than the other. Ectocentric, the centromere is close to the end such that one of the arms is very short and another is long. Telocentric, it is where the centromere is at the position. This chapter is being finished.